Good afternoon, dear viewers. I am Dr. Amit Bhattacharya, Associate Professor in Department of Zoology, Ramjas College, University of Delhi. Today I am going to present the first lecture in the biochemistry series, and the lecture is titled Understanding Enzymes, Structure and Classification. So in this particular module, we will be discussing about the basic understanding, what exactly the enzymes are, their classification, and some of the important functions which these enzymes does. Now, before starting, we should understand what exactly the enzymes are. So basically the enzymes are called as the biological catalysts. So they are a specialized group of protein which are present within the cells which performs the catalytic activity. So most of the enzymes are proteins, and basically they speed up the rate of specific chemical reaction within the cell without being destroyed or used up during this reaction. So they can be used over and over again. So if you see over here, the basic mechanism of the enzyme activity is there is an enzyme, and in the enzyme there are specialized sites which are called as the active site, to which the substrate binds, and subsequently the enzyme substrate complex is formed and the reaction proceeds to form the product which are released from the enzyme, and now this free enzyme can go back for another round of reaction. Now, in these cells, there are thousands and millions of different enzymes which are present, and each of these enzymes are characterized to perform specific activity or reactions within the cell. So the enzyme's mechanism is basically what we just discussed. Is there is a enzyme, free enzyme, so it binds to the substrate which is called as S, and subsequently it forms a transient molecule which is called as the enzyme substrate complex or the ES complex, and finally the product from this particular enzyme is released. Now in this particular reaction, the enzyme remains unchanged and can bind to another substrate in the subsequent reaction and can be reused several times within that particular cell. So they play a very important biocatalyst activity. Now the important question comes is, why exactly the enzymes are important to us? Now the enzymes are the key proteins which are performing several functional activities within the cell. So most of the activities which we are seeing within the cells are being performed with the help of these enzymes. So these enzymes plays a very important role. Now there are two important characteristic features these enzymes shows. One is the catalytic power and the other one is the specificity for the substrate. Now these catalysis generally takes place within a particular site where in the enzyme which are called as the enzyme active site, and nearly most of the enzymes which are known are in protein in nature. Now these enzymes, as I just told you, are specialized class of proteins which are responsible for catalyzing the various chemical reaction within the cells. So many a times in many of the disease conditions, when these enzymes acts abnormally or they don't act, so they becomes the ideal target for the drug therapies also. Now these enzymes are made up of proteins, or basically the amino acid chains. So the amino acids chain is brought together in a three-dimensional structure, in which we can see over here there are various amino acids which are brought together at a specific location, which is the enzyme active site. Now the protein structure has been ordered, or there is a hierarchy of the protein structures which we know. Now these are being classified into primary, secondary, tertiary and the quaternary structure. So understanding this is also very important for understanding the main structure of the enzyme. So the primary structure is basically the simplest level of the protein structure which is formed of the sequence of amino acids. So over here if we see, the example which is shown is the hormone insulin, which is shown over here. So there are two chains, A chain and the B chain, which are joined together with the help of the disulfide bond. So we can see there are three disulfide bonds which are there, and there is a protein amino acid sequence which is present in each of these chains. So each of these chains forms the first basic line which is called as the primary structure of the proteins. Now this primary structure further gets coiled up to form the secondary structure. Now these folded structures are formed basically with the help of various bonds, like the hydrogen bonds which are there. So there is an interaction between the various atoms of the backbone which forms the secondary structure. Now these secondary structure are basically classified into two major groups. One is called as the alpha helix, the other one is called as the beta pleated sheet. So over here we can see the right side image is showing the alpha helix structure, and on the left hand side we can see the beta pleated sheet which is shown over there. So these form the secondary structure, which are further complex structure from the primary structures. Now subsequently there is a tertiary structure which is formed, which is basically the three-dimensional structure of these secondary structures, which are further coiled and looped up, and these are called as the three-dimensional structure of the polypeptide, many a times referred as the tertiary structure. So we can see over here, with the help of the various bonds like the hydrogen bond, covalent bond and the other bonds, these particular structures are structured up to form the tertiary structure. So tertiary structure is basically formed with the help of the interaction of the R groups which are present in the amino acid, which makes up the protein chain over here. So the various R groups are shown over here. How these R groups are interacting to one another is shown over here in this diagram with the help of the various covalent and the non-covalent bonds over there. Now finally, the last order of the protein structure is the quaternary structure. So many of the proteins are made up of multi-polypeptide chains. So more than 2 or 3 polypeptide chains are present. For example, one of the common example is the hemoglobin. So these polypeptide chain, they interact with one another with the help of the bond, and each of these subunits come together to form the protein quaternary structure, which forms the final functional molecule within the cell. So the one of the best example is the hemoglobin over here. Now this is a diagram which shows the hierarchy of all these protein rearrangements, like the primary structure from the linear polypeptide chain, then comes the secondary structure which shows the alpha helix and the beta pleated sheets, then the quaternary structure, the tertiary structure, and finally the quaternary structure, which is formed with the help of the various subunits over there. Now the enzymes plays a very important role in the digestion functions within our body also. So some of the basic thing which is shown over here is the digestion of the protein, fats and the carbohydrates. We know that the proteins are basically made up of amino acids. So there are various aminopeptidases which are present within the body. What they do is they separate out all these amino acid structures from one another by breaking the peptide bonds within them. Then comes the fats, and the fats molecules are basically made up of the fatty acid on the glycerol backbone. So what these enzyme does is they separate out the glycerol backbone, and the fatty acids are released, which can be further broken down by various enzymatic reactions like the beta oxidation and the other reaction, and then the acetyl CoA formed from the beta oxidation can go into the citric acid cycle. And finally the carbohydrate molecule, we know that they are made up of sugar molecules, so it can be disaccharide, trisaccharide, polysaccharides like this one. So saccharide means the sugar molecule. So there can be glucose, fructose, galactose, maltose, lactose like this one, various mono and disaccharide. So there are various enzymes which is present within the body. The digestive enzyme, what they do is they break down these glycosidic bonds between them to release the free glucose molecules, and which can be further moving into the various metabolic pathway like the glycolysis pathway, which can further go into the Krebs cycle or the citric acid cycle for complete oxidation over there. Now some of the enzymes are active without the coenzymes, but majority of the enzymes, they need a coenzymes to be active. So when an enzyme is in an inactive state with the absence of the coenzyme, they are called as the apoenzymes. So when the coenzyme comes and binds to this apoenzyme, there is a conformational change which occurs within the enzyme, and now the enzyme is active and it is called as the holoenzyme. So there are two terms over here, one is apoenzyme, the other one is holoenzyme. So apoenzyme means an enzyme that is inactive in the absence of its coenzyme, while the holoenzyme is a complete enzyme where there is a apoenzyme plus the coenzyme which has activated the enzyme. So it is called as the holoenzyme over here. Now one of the example which is shown over here, I just discussed, is the apoenzyme plus the coenzyme forms the holoenzyme. Now over here I have shown an example which is of a enzyme lactate dehydrogenase. So this lactate dehydrogenase, what it does is it goes for a dehydrogenation reaction over here, Now the enzyme lactate dehydrogenase, it requires a coenzyme which is NADH. So in the presence of NADH it becomes active, then only the pyruvate will come and bind to it and it will form the enzyme substrate complex, and subsequently the product will be formed which is called as the lactate over here. By the release of the product and the NAD, the enzyme is now free for another round of reaction over there with the enzyme is lactate dehydrogenase. So this is a small example which shows that how exactly the apo enzyme and the coenzyme has to function together to form the active part, which is the holo enzyme, and go through the catalytic reaction. Now this is a diagram which shows how the apo enzyme, which is the inactive part of the protein enzyme part, how it binds with the non-protein portion, which is the cofactor or the coenzyme over there. As it binds, the enzyme becomes active to bind with the substrate over there. So the apo enzyme plus the cofactor or the coenzyme forms the holo enzyme over there. Now the cofactor can consist of the various inorganic ions such as the Fe3 plus, magnesium, Mn2 plus, zinc, like this one. So these inorganic ions forms the cofactors for the enzyme. Many a times there are complex organic molecules which are referred as the coenzymes for that particular enzyme. So many of the enzymes require both the types of cofactors, both the cofactors and the coenzymes also. Now the coenzymes are basically the organic compound that binds to the active side of the enzyme or near to it, which brings about certain changes in the enzyme and make it more active so that the reaction or the catalytic process can go further for forming the product over there. So many of the coenzymes are derived from the vitamin. So vitamin plays a very important role in our body because most of the coenzymes, which we will see in the subsequent slide, are being derived from the vitamins. So many of the coenzymes like the NAD plus, which is the nicotinamide adenine dinucleotide, it is derived from the niacin. Many of them are derived from the water soluble vitamins or vitamin B group, which acts as the coenzyme. So vitamins plays a very important role in the body. Now if we see the table over here, we can see the various coenzymes as well as the metal cofactors which are being shown over here. So if we see over here, like one of the most important enzyme is the hexokinase enzyme, which is one of the important enzyme in the glycolysis pathway. So we can see over here the hexokinase requires a metal ion, which is the magnesium, for its activity. So in the absence of that particular metal ion or the cofactor, the enzyme won't be active and the reaction won't proceed further. In the case of the enzyme coenzymes of the enzyme, so we can see over here the pyruvate dehydrogenase, the important coenzyme is the thymine pyrophosphate, which is there. Then the biotin is there. There is very tetrahydrofolate is there. So we can see many of the coenzymes have been derived from the vitamin. So vitamin plays a very important role in helping giving us many of the coenzymes within the body. Now before going into the enzyme kinetics, let's discuss some of the important examples which we see over the time. Now one of the example which is shown over here, one of the enzymatic activities is shown over here is the fireflies. So we know that the fireflies were in the night, when we see the fireflies, the abdominal area of the fireflies are glowing. Now why exactly that glow is occurring? So what the firefly does is they produces a chemical reaction inside their body which allows them to light up. Now what happens is the substrate, which is the luciferin, in the presence of the enzyme which is the luciferase enzyme and magnesium, it goes for a processing part using the ATP molecule to produce light. Now once the light is produced, the abdominal part glows up and the product which is formed is called as the oxyluciferin over here, releasing the PPI or the pyrophosphate and the AMP molecule. So this is the example, a simple example of showing how exactly the enzymatic activities are playing a major role over here. Now the other example is the jellyfish. So we know that the jellyfish, if we watch the jellyfish, they glow. So many a times they emit out the bioluminescence of blue light over there. So this is again because of a enzymatic reaction which is called as the enzyme is called as the aquaporin. So we can see over here, the aquaporin is there. Now in the presence of the enzyme aquaporin, the enzymatic reaction takes place. So the substrate binds to the enzyme, forming the product, and the light is released over there. Now once the light is released, the jelly fish, they start glowing over there. So for this pioneer work, these three scientists were given the Nobel Prize for discovering and development of this green fluorescence protein within this jellyfishes. Now what they did is they started studying the glow within the jellyfishes in the early 1960s. Now what they did is they isolated the bioluminescent protein from these particular jellyfishes, and they found that there was a glow of the link blue light which was given up, but subsequently the jellyfishes glow green. So they found that there is a protein which is there. What is doing is this is absorbing the blue light and giving a green light over there. So later on it was named as the green fluorescence protein, which was emitting the green light within them. Now for this particular work, these scientists were given the Nobel Prizes. So we can see how important the enzymes plays an important part in our day-to-day activity and the various biological activities present within the animal kingdom. Now the enzymes are characterized by various important points. Now there are four important characteristics which are there for the enzyme. The first is the enzyme catalysis, which is for the rate enhancement. So most of the enzyme, what they do is they enhance the rate by lowering the activation energy, and the catalysis increases. Now the next important thing is that the enzymes have got a specificity for the substrate, so it varies from substrate to substrate. The third important characteristic of the enzyme is the regulation of the enzyme reaction. So the enzymes are regulated by various ways. So there are various feedback mechanisms which are present. Then there is allosteric modifications which are there, through which the enzymes are modulated and they are regulated. And the fourth important point is the enzyme, most of the enzymes, they require the cofactors or the coenzymes for their functioning and the activation part. Now over here, this particular the reaction shows what the enzymatic work. So the free enzyme, it binds to the substrate to form the enzyme substrate complex. Then the catalytic step start taking place, and finally the enzyme and the product is formed. So ES complex is many a times called as a transient state molecule, because it can go back to form the enzyme and the substrate, or it can further move into the catalytic step to form the enzyme and the product over there. Now this particular image also shows how the enzyme works over there. So we can see over here there is an active site to which the substrate binds, and as the substrate binds, there is an enzymatic reaction which takes place, releasing the product, and finally the product is released. So the enzyme moves back to its original position for another round of reaction over there. Now enzyme active site, again we discussed, is there are certain grooves or the pockets which are present within the protein structure. So we can see a diagram over here where there are protein grooves which are present, in which the substrate, which is shown yellow in color, the binds in a proper orientation for the reaction processes. Now the enzyme active site is made up of various amino acid, but when we stretch out these amino acid polypeptide chain, we find that all these amino acid are scattered at various locations within that polypeptide chain. So what happens is, because of the three-dimensional folding, all these amino acids which are present at various locations, they come to a near point and forms a specialized groove or the enzyme active site, and when it is enhanced, they are present at various locations in the primary structure. So this particular diagram is showing how exactly, if the three-dimensional structure or the ribbon structure is showing the enzyme active site, when this particular enzyme polypeptide was formed into a primary structure, so the polypeptide chain was elongated, and we find that the various amino acid at various positions, like over here it's shown 35, 52, like this one, they came into a single point to form a specialized groove over there, which was forming the enzyme active site. Now this is again another example of the enzyme substrate complex, which is shown over here, how the substrate goes and binds to the enzyme active site. Now the functioning of the enzyme was majorly given up by two model systems. Initially there was a model system which was given for the enzyme kinetics, which was called as the lock and key method model system. Now this was given by a scientist, his name was Emil Fischer, in the year 1894. Now in the 1894 what he proposed is the enzyme has got a characteristic specialized location in which the substrate comes and binds. So the substrate is having a complementary structure to this enzyme active site, so it goes and binds to that particular location like a key. So the lock is basically the enzyme and the key is the substrate, so it goes and binds and the reaction starts with the help of forming the enzyme substrate complex, and finally the enzyme plus the product is formed over there. Now subsequently this model system was not recognized or was not accepted because it was a rigid model system. It doesn't discuss about the enzyme flexibility, conformational changes, because which is given up by the various polypeptide or the amino acids within the active site. So subsequently another model system came up which is one of the most accepted model system. It is called as the induced fit model system. It was given by a scientist called as Koshland in the year 1963. Now what he proposed is the substrate, when it comes near the enzyme, what happens is the enzyme, there is a conformational change. So the enzyme active site is not in a complementary structure to the substrate, but as soon as the reaction initiates, there is a conformational change within the enzyme which occurs, due to which the enzyme active site now gets or retain into a complementary structure to the substrate, and finally the substrate binds to it and the enzyme substrate complex is formed and the reaction goes about. So this particular model system, it talks about the induced fit. So as the name is suggesting, induce means the substrate coming by is inducing the enzyme to complement its active site to the substrate, and once the active site is complemented, the substrate comes and binds and finally the enzyme substrate complex is formed over there. Now this is a basic diagram which shows the enzyme kinetics. So as I told you that the enzyme works in a very simple process, that it lowers the activation energy. So if you see the diagram over here, this is a diagram which is plotted with the help of the energy and the reaction process. So initially there was a reactant, so the substrate came, the enzyme came and the substrate was there, so they complemented to each other, the enzyme substrate complex was formed and the product was formed. Because of the presence of the enzyme, we show that the catalysis activation energy lowered up, and finally the graph which is shown in blue color is the one. So there is a activation energy which has lowered up with the catalysis, and the enzymatic reaction occurred in a much more faster way. So this gives an idea that how enzyme helps in the lowering of the activation energy and catalysis of the reaction in a much more speeded way. So there are various reasons by which it does is, firstly the activation energy is lowered because of the reduction of the amount of energy which is needed to complete the transition, and the orientation also plays a very important role. So in this part of the discussion, we discussed about the basically what exactly the enzymes are, the definition of the enzymes, and then we discussed about the two important model systems, the Koshland model system, which is the induced fit, and the lock and key model system, which talks about the lock and key. And finally we discussed about the enzyme kinetics, in which how the lowering of the activation energy is helping the enzymatic reaction to fasten without using the enzyme itself or without distorting the enzyme for the subsequent reaction. So with this note, I come to the end of the first part of the lecture, and in the next part we will discuss about the enzyme kinetics, the various inhibition model systems, the inhibitions, the competitive inhibition, non-competitive, and the various systems, as well as the Michaelis-Menten reaction, where we will discuss about what is Vmax, what is Km value, and how exactly it is extrapolated to describe a enzyme kinetics in a better way. So with this note, I come to the end of the first part. Thank you very much. Thank you.